Hello guys, so now we shall be continuing the next part on. So we have discussed about incineration, right? So we have already discussed that we have got two types of heat, dry heat as well as the moist heat, and in the dry heat part we have discussed the flaming, and we have also discussed incineration. Now we shall be focusing on the hot air oven. Now what is this hot air oven? Okay, so what is this hot air oven? Now in this hot air oven, few things you need to understand, that we also have something called autoclave, right? So where did I write autoclave? I have written autoclave in the moist heat. You use more than 100 degrees, so that is the autoclave. Basically for sterilization we mainly use autoclave. Okay, that is the most commonly used. Second most commonly used is this hot air oven. Okay, so if someone asks you what is the second most commonly used device for the sterilization and disinfection, is your hot air oven. So this is gonna be second most commonly used after what? After autoclave. Second most commonly used after autoclave. Okay, now in this hot air oven, basically you maintain three temperatures at three different timings. Okay, so what are those temperatures are? You heat the things, I mean, you know, surgical instruments and all. I will be writing what all things which you basically disinfect and sterilize in case of hot air oven. But one thing is that three different temperatures you use. One is 150 degrees centigrade, next is 160 degrees centigrade, next is 170 degrees centigrade. So it is easy, 150, 160, 170. Okay, but now the timings are little bit different. You heat at 150 degrees centigrade for about 115 minutes, 115 minutes. You heat at 160 degrees centigrade for about, let us say, 1 hour. 1 hour is how much? 60 minutes. You heat at 170 degrees centigrade for 40 minutes. 40 minutes. Okay, so how do you remember this, guys? So the temperature is increasing. As the temperature is increasing, I told you, you have to decrease the time, right? So lowest temperature is 150, which has got the highest time, and highest temperature is 170, which has got the least time. Okay, so in this way you have to remember it. So these are the three different temperatures. Okay, fine. So what are you going to sterilize and disinfect here? The things which the materials which I am going to sterilize and disinfect are, one is, let us say, glass flask. In the lab you use glass flask, right? You use glass flask in the lab. You also use test tubes in the lab, right? So glass flask, test tubes, right? Apart from test tube, what do you use? Pipettes, petri dishes, right? So you use petri dish, pipettes, and apart from that, another important thing is that the sharpeners, whatever are there, either it might be a scissor, or it might be a scalpel, or let us say it can be a forceps. So all these are called as sharpeners, right? The surgical instruments. So sharp surgical instruments, sharp surgicals. What are the sharp surgicals? Sharp surgicals are mostly three. One is scissors, right? Forceps, as well as scalpels. So even them you scalpels, right? So even the scalpels, I mean, even these things you basically sterilize and disinfect. So these are the important things to remember. Okay, now in hot air oven, another important thing is that you have to first of all see whether this hot air oven is working or not. I mean, you have to check the efficacy, isn't it? You have to check whether this hot air oven is working enough to disinfect all these things or not, whether it is working or not. You have to check the efficacy. So how do you check the efficacy? You check the efficacy by three ways. Okay, so how do you check the efficacy? The efficacy of this hot air oven is checked by three ways. One is called as your physical way, the next one will be chemical way, and obviously the last one will be biological way. So in the physical way, what are you going to do? What are you going to do is that whether this machine's time, display, temperature, all of them are working or not, you have to check. Okay, so in the physical method, you check the display, you check the temperature, right? I mean, you basically display the temperature and time. Temperature and time. Second thing is chemical. Chemical method of checking the efficacy is you basically use a special tube called as Brownies tube. Okay, Brownies tube. So this is also called as Brownies tube number 3. Okay, now when it comes to the biological, you have to check it, right? So what you do is that you keep some spores of the bacteria, or you put the bacteria itself and you start heating at these degrees and look whether the bacteria is dead or not. I mean, the spores are denatured or not. So if they are denatured, it means it is working. Okay, so but spores of which bacteria? We basically use Bacillus subtilis, or we use Bacillus atrophius. So basically you use the spores of these bacteria. Okay, Bacillus subtilis and atrophius, you use the spores of this bacteria to check the efficacy. Okay, so this will be your biological method. So we are done with what, guys? We are done with the third important thing, that is hot air oven. Now we shall be switching on to the next important thing, that is moist heat. Now in the moist heat, I already told you that you use 3 different temperatures. One is called as less than 100 degrees, equivalent to 100 degrees, and more than 100 degrees, right? So if you are using less than 100 degrees, there are different methods where you use a moist heat of less than 100 degrees to sterilize and disinfect. So one is pasteurization, serum bath, vaccine bath, as well as incineration. Okay, so when it comes to pasteurization, I have already talked previously long back about this pasteurization in the previous lectures, but anyways, let us discuss it again. That we are basically discussing what? We are basically discussing a special type of heat that is called as moist heat. In the moist heat, we can use 3 different temperatures. Now I am mainly discussing on less than 100 degrees centigrade temperature. Now in that less than 100 degrees centigrade temperature, the first thing which I have taken is pasteurization. What is pasteurization once again? Yeah, what is pasteurization, guys? Pasteurization means either it might be a packed milk or an unpacked milk, right? You are destroying all the microorganisms which are located in the milk. So to destroy that, you have to heat the milk. Okay, only then you can destroy these microorganisms. So after destruction of the, why are you doing this? Why are you doing this is that you wanted to increase the shelf life of the milk. To increase the shelf life of the milk, you have to kill the microorganisms, or else microorganisms will completely damage the milk. Okay, so we use pasteurization. So the pasteurization of milk is simply used for disinfection, right? What is pasteurization? Pasteurization of milk is nothing but disinfection of milk. I hope you understood now. Okay, now in this pasteurization, you basically use 3 different temperatures. This is first temperature, this is the second temperature, this is the third temperature. Now at 3 ways you can heat it. 3 ways you can heat it. What is the first way is that in the first way you use a temperature of about 63 degrees centigrade, and after that in the second way you slightly increase it. You increase it to 72 degrees centigrade. Okay, so how do you remember? After 6 what comes? 7. Next 3 minus 1 is 2. So 63 degrees and 72 degrees. Next what you do is that you heat it at 149 degrees centigrade. And what did I tell you again? I told you that whenever you are increasing the temperature, you have to decrease the time, right? So 63 degrees centigrade, you are heating it for, let us say, 30 minutes. 72 degrees, you have to decrease the time. So you will decrease it to, let us say, 15 to 20 seconds, and 149 should be for 0.5 seconds. Okay, 0.5 seconds. So you have to name them, right? So this method, the second method as well as the third method. Okay, so the first method you call this method as holder method. You call it as a holder method of pasteurization of milk. The next method is called as flash method. And third one is you are increasing it to 149 degrees centigrade, where the temperature is too high, right? So this is called as ultra high temperature method. Ultra high temperature method. So these are the three ways through which you can disinfect the milk. Okay, fine. So you have proposed these three ways. You told someone that through holder method, flash method and ultra high temperature method, I am going to kill down all the microorganisms within the milk and increase the shelf life of the milk, so that you can store the milk for a longer time. Now you will ask me, I don't want to waste my milk based upon your experiments. So can you please prove me that you really experimented all this and these experiments are really working? Can you please prove me? So how do I prove? How do I show the efficacy of pasteurization? How do I prove that this pasteurization technique is a real technique and it's working? How do I prove that? So how do I prove is, how do I show efficacy of pasteurization? Yeah. So how do I show the efficacy of pasteurization? I do two tests and I'll prove you that this pasteurization technique is working. Now one test, you call it as coliform test. Coliform test. Coliform test in the sense, basically in the milk you have got some bacteria, guys. You have got some bacteria in the milk. These bacteria are called as coliforms. Okay, so what are these bacteria basically in the milk which you see? One is called as E. coli, Escherichia coli. Another one is your Enterobacter, E. coli, Enterobacter, as well as your Klebsiella. So these three bacteria, you call them as coliforms. Coliforms, which are basically present in the milk, right? Definitely you will see them in the milk. Now if I use one of these three methods and try to kill all of them, it means I have proven what? I have proven that these methods of pasteurization are effective, isn't it? These methods of pasteurization are working. So I have to prove now, I have to kill them. So now what I do is that I will take a milk. In this milk all these coliforms are there. Now I start heating either by holder method, flash method or ultra high temperature method. Now after heating it, I am telling you that all these bacteria are dead. Now I have to prove it. How will I prove is that, in normally you take a milk, guys, you take the milk, don't heat it, don't do anything, don't pasteurize it. What you do is that you simply pour that milk on McConkey agar, okay, and keep that aside for one complete night. In the morning, either you will see that entire agar turning into acid or gas. Why? Because bacteria release acid, bacteria produce gas. Okay. Now you have taken the milk, you have pasteurized it, you have heated it. Now what you are doing is that and you are telling that now the bacteria in this pasteurized milk are dead, and you add that milk onto the McConkey agar, okay, and put it overnight. So in the next day morning when you come and see, if acid or gas is not produced, it means the bacteria are dead. So in this way, can I prove it that the methods are working? Yeah. So what I do is that I take the milk sample, right? I take the milk sample and add this milk sample to McConkey agar. After adding the milk sample to Mekong ki agar, I will put it overnight. I will put it overnight. And what should I look for? I need to look for acid or gas. Right? So if there is no acid or gas, it means these methods are working. Clear? So this is a test which you gonna do. Right. I can also do another test also, I told you that. So what is this test is that the name of this test is phosphatase test. That is phosphatase test. Phosphatase test. Now what is this phosphatase test? What is this phosphatase test is that normally in the milk you have got this enzyme called phosphatase. Okay. So what you do is that you take a container, you add milk into that. I told you milk has got an enzyme called phosphatase, right? It means within this container of milk there is phosphatase. Now you add diphenyl phosphate, DPP, diphenyl phosphate. Now when you add diphenyl phosphate, what will happen is that this diphenyl phosphate, right, DPP, diphenyl phosphate, let me write it. So this diphenyl phosphate, with the help of this phosphatase enzyme, diphenyl phosphate will completely change the color of the milk. So because of this, the milk color changes. Milk color changes. But if I am pasteurizing the milk, what will happen is that this phosphatase enzyme is lost, right? It is denatured. So enzymes are made up of proteins, right? So because of this high heat, the proteins are damaged and the enzyme is denatured. So what will happen when the enzyme is denatured? Even after adding diphenyl phosphate, the milk color doesn't change. So even by this, I can prove that the pasteurization techniques are working. Clear? Right. So we have discussed, guys, right? Let us go back to that chart which I have mentioned before, that in the moist heat, the first important thing which you have to discuss was pasteurization, and we are done with this pasteurization. Now we are switching on to the next important thing, that is serum bath and next the vaccine bath. Okay. So what is your concept of serum bath or vaccine bath? Now, what is serum bath? What is serum bath? Now, in this serum bath, first of all, let me discuss the temperatures. You use a temperature of about 56 degrees centigrade for about 1 hour. 56 degrees centigrade for about 1 hour. Now, what are the materials you basically use in this serum bath? What are the materials you basically use are any kind of serum or body fluids. Okay. So, any kind of serum or body fluids, because why? What are you doing here? See, any kind of serum or body fluids can also get infected, right? So they are the high source of infection. So what you are doing is that you are heating the serum or body fluids so that within this there are some proteins. So these proteins will be damaged. So what will happen is that within this serum or body fluids you have got coagulable proteins. Coagulable proteins, and these proteins will be damaged. So in this way you can completely sterilize the serum or any kind of body fluids. Okay, so that is called serum bath, pretty simple one. And the next one is, after serum bath we have got the vaccine bath. We have got something called vaccine bath. Now what is this vaccine bath? Vaccine bath in the sense, see, what is the basic, what is vaccine, guys? Vaccine is nothing but you take the microorganisms, you inactivate it, right, and then inject it into the body. So this is what is called as vaccine, right? For example, let us say polio vaccine. In polio vaccine, what are you gonna do? What are you gonna do is that you gonna take the polio virus, you will inactivate it first and then inject it into the body, right? So that your body will produce some antibodies, memory T cells and all, memory antibodies and all against that. Okay, so this is the main concept of vaccine. But if they are activated and you are injecting that polio, that can harm you. So that is the reason why you inactivate it. So how are you going to inactivation is also called as attenuation. That is why you call it as live attenuated vaccines and all. You know it, right? So how do you attenuate it? How do you inactivate those polio virus? You inactivate it by using this vaccine bath. What you do is that you take the vaccine and you heat it at 60 degrees centigrade for 1 hour. Okay, and then what will happen? The organisms within that vaccine, they get inactivated and then you can use it happily, right? So for preparation of vaccines, you basically use this vaccine bath, right? So we are done with what? We are done with the heat part. Okay, so we are done with the serum bath, we have done with the vaccine bath. So what is your concept of inspissation? Right? So in inspissation, you can see the first two letters I and N. I and N means intermittent, right? So inspissation is intermittent heating, intermittent sterilization. So you call this also as intermittent sterilization. Intermittent sterilization. Now in this intermittent sterilization, first of all you have to lower the temperatures. The temperatures are, what you do is that, first of all what do you heat here is, either it might be a serum or egg containing culture media. Okay, so you basically heat the serum and egg containing culture media. So what do you do is that you intermittently heat it. What do you mean by intermittent heating is that you take the serum or egg containing culture media, you heat it at 80 to 85 degrees centigrade. Okay, you stop it. Next day again you heat it at 80 to 85 degrees centigrade, and again third day also you heat it at 80 to 85 degrees centigrade, and now you can tell that we have done intermittent sterilization and completely it is sterilized, right? So when you are heating it, if there are any kind of spores within this serum or egg containing culture media, if there are any kind of spores, on the first day heating the spores are not damaged, on the second day heating the spores are not damaged, but on the third day heating the spores will be damaged. So that is the reason why you heat it for 3 days. That is why you call it as intermittent heating. Okay, so what is this intermittent heating? You use a temperature of 80 to 85 degrees centigrade for about 30 minutes daily. 30 minutes daily, and for how many days? You use it for 3 days. If there are any spores, they are going to die on the third day. Okay, so this is called as inspissation. Now next important thing is tyndallization. Now what is your concept of tyndallization? But before discussing tyndallization, one thing you have to be sure that we have discussed everything. I mean, we have discussed pasteurization, we have discussed inspissation, we have discussed serum bath, vaccine bath. So in all of them, if you look at the temperatures, they are less than 100 degrees, right? So here also that is less than 100 degrees, less than 100 degrees, right? So in all of them, you are using a temperature of less than 100 degrees. But now you are switching on to the next one where you are using the temperature equivalent to 100 degrees. Temperature equivalent to 100 degrees centigrade. So what is that method where you use the temperature of equivalent to 100 degrees centigrade? That is tyndallization. That is what? That is your tyndallization. Now in tyndallization, what is the temperature which you use? I told you, right, 100 degrees. So you use a temperature of 100 degrees centigrade, and for how many days do you use? You use it for 3 days. So 20 minutes. daily you heat it for 20 minutes for 3 days, right? So completely for 3 days. So daily I'm heating it 100 degrees for 20 minutes for 3 days continuously, right? So can I also call this sterilization is also a form of intermittent sterilization? Yeah. Previously, inspiration is called intermittent sterilization. So I am also calling this one as intermittent sterilization. Intermittent sterilization. Intermittent sterilization. So previously in inspiration I was using serum and egg containing culture media, right? So even here also I use the same serum and egg containing culture media. So what I use? Serum and egg culture media. Egg culture media, right? So at 100 degrees there is only one method, that is sterilization, and now we are switching on to the next one where we are sterilizing at more than 100 degrees centigrade. So in more than 100 degrees centigrade, what was the thing, guys, which we basically studied there? That was your autoclave. Autoclave. So autoclave, I told you, autoclave is the most commonly used procedure, right? The most commonly used machine. And what is the second most commonly used? I told you already, that is hot air oven. Okay, so autoclave is most commonly used. Okay, it is most commonly used. You use three different temperatures. What are the three different temperatures? 121 degrees centigrade, then you increase it again 126 degrees centigrade, then you increase it again 132 degrees centigrade. I told you, as the temperature is increasing, you have to decrease the time. So 121 is the least temperature, and you use the time limit of about 15 minutes. For 15 minutes at 121 degrees centigrade, you heat it, right? So this autoclave works like this. So in 126 degrees centigrade, you have to decrease the time, right? Because you have increased the temperature. Let us say I have decreased it to 10 minutes. And finally, it will be less than 10 minutes, right? That is 3 minutes. And another important thing is that keep in mind, as the temperature is increasing, the pressure also increases. So at 121 degrees centigrade, let us say the pressure is about 15 pounds. 15 pounds psi. Then as the temperature is increasing, the pressure also should increase. Then the next one will be 20 pounds. And the next one will be 30 pounds psi. So this is a table which you have to basically remember, guys. Okay, very, very important here. Right. Now another important thing is that, okay, fine. So you are telling that in autoclave we use these three different temperatures, but on what materials do you use these three? So the materials are, you can autoclave any culture media. Okay, any culture media you can autoclave and kill down all the spores in that culture media, but except egg and what previously we have discussed, serum and egg containing culture media, because for serum and egg containing culture media we have special procedures like sterilization and inspiration. We don't use this autoclave. Okay, so autoclave is used for all culture medias except this serum and egg containing culture media. So you use it for all culture media except for serum and egg containing culture media, right? Now the next important thing is that you can use any surgical instrument it might be, you can autoclave it. Okay, so within that autoclave machine you can keep any surgical instrument other than the sharpeners. Except sharpeners, you can keep any other surgical instruments. So all surgical instruments except sharp materials, you can use anything. Third important thing is that you can even autoclave any kind of plastic or rubber material, right? You can even autoclave any kind of plastic or rubber material. What is your plastic or rubber material? What medical equipment is having plastic or rubber, guys? Tell me. Gloves, right? Surgical gloves. Not only surgical gloves, what is the next thing? The Foley's catheter, right? So Ryle's tube. So all these are the plastic or the rubber material. So even you can use. Okay. So this is all you need to know about the sterilization and disinfection techniques, guys. So thank you so much for watching my lecture. Goodbye.